Yes, thank you, Jordi. First of all, apologies. I'm currently in my office and no webcam, but I suppose that was not a problem. It is not a problem. Thank you on behalf of all our authors for your kind invitation. I will present the most important results. I think this is very important paper, and we believe it will attract readerships. Our aim was to select and present the most important circulating biomarkers for vascular aging, as well as underlying pathophysiological mechanisms. Next slide, please. With this purpose, thank you. With this purpose, we searched the Medline base for a period of 32 years. And the keywords we used were circulating biomarkers, cardiovascular disease, aging, and vascular aging. Initially, we selected 30 biomarkers, which were based on criteria introduced by Vlachopoulos and others. We applied the next inclusion and exclusion criteria by selecting most important circulating biomarkers. Inclusion criteria were first studies investigating the association of a biomarker with cardiovascular disease and vascular aging. Secondly, studies using blood, serum, or plasma for biomarker analysis. And finally, we include those papers which cover this period that I mentioned from January 1989 until October 2021. Those were peer-reviewed articles and all types of reviews published in English, and we exclude unpublished thesis reports and conference proceedings. Next, please. By applied these stated criteria to the literature, we identified initially 30 biomarkers, but this list, after refinement, resulted in a short list of circulating biomarkers containing 8 of them, which are oxidative stress-based superoxidase mutase, but after our reviewer's suggestion, we include more molecules which are oxidative stress-based, such as catalase, glutathione peroxidase, etc. Inflammation-based, we include high-sensitivity C-reactive protein and interleukin-6. Cellular matrix-based, we include several classes of matrix metalloproteinases, which we described in details, and the growth differentiation factor 15, among many others, which are important in vascular aging. And from epigenetic based, we include microribonucleic acids, several classes were described, DNA methylation, and telomere length. We present the data for those 8 selected biomarkers according to next sections. They are pathophysiology, association with physiological aging and vascular markers, association with cardiovascular disease, and finally, we include measurement method for each of 8 selected biomarkers. Next, please. This figure illustrates those we selected. 8 biomarkers are presented here with high potential to be used as biomarkers of vascular aging, or possibility to be used, or they are already in use as a circulating biomarkers, and the underlying pathophysiological mechanisms. Next, please. Reactive oxygen and nitrogen species are very important in normal functioning of human body, and they have, of course, dual role in an organism. They are small molecules, ions, or radical species, which are important in cell signaling and development under the physiological conditions. Regarding the vascular system, they are essential for normal vascular functions, including, let's say, endothelial homeostasis as the most important, and smooth muscle cell contraction. These species contribute by several different mechanisms to vascular aging. First of all, we think this is most important, vascular remodeling by inducing proliferation and migration of vascular smooth muscle cells, vascular cell damage, and consequently the recruitment of inflammatory cells. Finally, there are changes caused by activity of these species on biomarker molecule structure and function. First of all, lipid peroxidation, activation of metalloproteinases, and finally deposition of important components of extracellular matrix. We include in our review paper more data. For example, we include antioxidative mechanism for reducing excess of reactive oxygen and nitrogen species, enzymatic and non-enzymatic sources of these species. But finally, we may conclude that exact mechanism of aging caused by oxidative stress is still unclear, but there is no doubt that increased production of these species, both oxygen and nitrogen is observed with aging. Next, please. Aging is a characteristic of chronic sterile low-grade inflammation. We're discussing in this chapter in more detail inflammaging, which is considered as a pre-status mechanism for many age-associated disease, including cardiovascular disease, which we are focused on. There are many inflammatory factors, but we think that the key molecules associated with inflammaging and aging are increased levels of pro-inflammatory cytokines, first of all, interleukin-6 and C-reactive protein. Interleukin-6 is very sensitive biomarker of low-grade inflammation, and according to literature, we may propose it as a representative biomarker of vascular aging. High sensitivity CRP is an acute phase reactant. This molecule is a non-specific inflammatory biomarker. Its production is stimulated in response to inflammatory mediators, including those two interleukin cytokines, and its level increase in age-dependent manner across aging elderly population without evident cardiovascular diseases. Next, please. Cell matrix is a very important component of each cell, containing many different proteins which are essential for normal function of cells. Cell matrix contains collagen, elastin, fibronectin, etc., and all they represent nice substrates and appropriate substrates for activity of this class of enzymes. Matrix metalloproteinases belongs to the class of proteolytic enzymes. They are multi-domain, calcium-dependent, and zinc-containing peptidases. They are activated by some inflammatory signaling, and as a result, they are able to degrade these molecules, which are essential components of extracellular matrix. They have very important role in aspects of aging, hypertension, and atherosclerosis within the arterial wall. And once activated, they cause arterial remodeling by some of next stated mechanisms, pathophysiological mechanisms, such as endothelial inflammation, intimal medial thickening, elastin fiber network destruction, and finally arterial fibrosis. As I said, we described many members of this very important family of proteolytic enzymes. Among them, we propose the activity, increase the amount and activity of these matrix metalloproteinases 2, 7, 9, and 14 in the aortic wall. And matrix metalloproteinase 9, its activity is positively correlated with aortic and brachial pulse wave velocity in patients with hypertension, including those with isolated systolic hypertension. Regarding the matrix metalloproteinase 2, gelatinase A, and MMP3 or stromatolizine, it has been well documented that they are associated with central arterial stiffness parameters in patients with diabetes mellitus type 2. And finally, matrix metalloproteinase 1 level shows positive correlation with both pulse wave velocity and augmentation index. And finally, we conclude that despite many controversy that still remain to be resolved, there is no doubt that these enzymes are tightly connected with changes of vascular wall, and they are very important in vascular pathophysiology. They may have a prognostic value to be used as a biomarker in the vascular aging. Next, please. Among many growth differentiation factors, we choose that one. GDF15 is a member of superfamily of cytokine superfamily of transforming growth factor beta, and it has been shown that GDF15 levels rise with aging, and they exhibit, this molecule exhibits the strongest positive association with age. Molecule is directly linked to inflammaging. Molecule is produced, is colocalized in macrophages, and it has been identified in human carotid atherosclerotic plaques. It has a dual role in vascular biology. It can exert either pro or anti-inflammatory, anti-angiogenic and apoptotic effects. And it has been shown that GDF15 may play an important role in the initiation and progress of atherosclerosis. And finally, conclusion regarding this molecule was that it has a strong predictive ability of clinical outcomes across different cardiovascular risk populations. Next, please. Among epigenetic-based circulating biomarkers, which represent the modification of influencing gene expression independently of gene sequence, we include DNA methylation, microribonucleic acid, and telomere length. Firstly, we described many classes of microribonucleic acids. It has been shown that vascular aging is strongly associated with the dysregulation in the expression of different microRNA, which are involved in crucial pathogenetic mechanisms. They led to next pathophysiological mechanisms and changes, impairment of angiogenic processes, decreased cellular stress, and plaque formation, destabilization, and finally plaque rupture. We focused our attention on several classes of these very important molecules, such as MIR1 and 133, which have been well documented correlated with arterial markers of subclinical atherosclerosis, and circulating microRNA A21 and 133B. They were dysregulated in patients with coronary artery disease without myocardial infarction. Next, please. DNA methylation is an enzymatic process which is in fact a transfer of methyl group from adenylmethionine to the 5th carbon atom to cytosine. It has been shown that there is almost 2% of the modification of the methylation, including CPG sites in DNA structure. But after discussion, very thoroughly, we discussed the role of DNA methylation. Our conclusion was that no human data regarding the association of DNA methylation with markers of vascular stiffness and DNA methylation-based measures were found to predict all-cause mortality over and above chronological age and traditional risk factors. Next, please. Regarding telomere length, we know that they represent non-coding repetitive DNA sequences containing 6 bases, and they are found at each end of the chromosomes of eukaryotic cells. They are highly conserved, and they have length from 11 to 15 kilobases. They are under investigation in the last several years, and it has been shown and well documented in recent years that functionally they preserve the genomic integrity by acting as protective caps to the genetic material and preventing attrition. And in age, regarding the aging, they are significant circulating biomarkers, but there is no consistency across different sexes and aging groups so far. Next, please. Our review paper includes 250 references. We provide 2 tables and 2 tables which are supplementary material, in which we provide extensive list of circulating biomarkers related to vascular aging, which are most important, and in table 2, which are less important. Next, please. Instead of conclusion, to avoid extensive conclusion, the main message of our paper is stated here. The new paradigm, this is our proposal, the new paradigm is the development of diagnostic panels of biomarkers. In this process, we must progress our understanding of existing biomarkers in addition to identifying new biomarkers and evaluate their real clinical relevance. This is very important, in addition to embracing new technologies in the fields of omics, including proteomics, glycomics, lipidomics, metabolomics, and metalomics, especially, and bioinformatics tools to achieve this. And next, please. We thank all our authors and reviewers for very useful comments and suggestions. We thank the core group members. Our driving force, I must say, driving force was Dimitrios. He's not present here, but all authors did great job, I think. Thank you very much for your attention.